Claudio has introduced some of these topics. Let me put some numbers on some of the scenarios that were alluded to. My disclosures are shown here, and I am obviously going to talk about products that the companies make. Probably more importantly, I have to disclose that I've taken a number of cappuccinos with Claudio, but this seems to not be uncommon. In fact, as you go around the world, you see various historical figures, both living and dead, appear to have visited with Dr. Ronco. I thought you were going to quote Mao Zedong, not Winston Churchill, which is why I had so I have to get the Winston Churchill slide queued up the next time I follow you. That might be too inside of a joke. Only you and I really understand what this is really about, but that's okay. That's okay. All right. So one of the things we have to ask is who to treat. Remember that rates of AKI are quite high. 50 to 60% of ICU patients develop AKI, but they don't all develop AKI at the same time. 30 to 35% of patients with pneumonia develop AKI, but not every pneumonia patient is the same. Patients who have septic shock, even higher, 65 to 75%. Patients undergoing cardiac surgery, 55 to 70% of patients may develop acute kidney injury. However, AKI stage 1 is not always associated with adverse outcomes. Clearly is in the case of sepsis, not so clear in the case of cardiac surgery. Isolated oliguria is not always predictive. For example, in cardiopulmonary bypass, because the perfusionists often dehydrate the patient at the end of the case so much that isolated oliguria may not be as predictive. Most AKI is already present at admission in the case of sepsis, and so enrolling those patients in trials may be more challenging. Only 2/3 of cardiac surgery patients have AKI within the first 48 hours of surgery, and late AKI is probably attributable to complications of surgery rather than the surgery itself. And so you need to think about enrichment strategies, and there are various strategies that can be used. This is the cardiac surgery data that Alex Zarbach and I have been working on. This is from about 6,000 patients undergoing cardiac surgery. This isn't published yet, but in fact, it hasn't even been submitted yet, so you're practically the first to see it. And what I'm showing you here is the rate of major adverse kidney events in 6 months. And this is major adverse kidney events is death, dialysis, or persistent renal dysfunction, doubling of serum creatinine at 6 months. And what I'm showing you here is the rate of MAKE based on whether you have no acute kidney injury by creatinine or urine output, which is shown here. And of course, it's quite low, and this is all sort of driven by death. It goes up slightly if you just have a little bit of transient oliguria. This is no AKI by serum creatinine. This is AKI stage 1 by urine output. This is stage 2 by urine output. This is actually statistically significant, but I defy you to tell me that you could enroll enough patients in a clinical trial to show a significant impact. When you start to get into the yellow and indeed into the orange, the rates soar, and by the time you're up here, where you certainly have stage 3 by both creatinine and urine output, the outcomes are terrible. In fact, just death or dialysis, just death is so high in this population that this is worse than most of the cancers that we treat, which, by the way, parenthetically, is why I get very upset when people tell me that they can't afford biomarkers when we're easily willing to spend hundreds of thousands of dollars to preserve 6 months of life for patients with various forms of cancer. I'm not suggesting that that life isn't valuable. I'm suggesting that there's certainly no parity with what we're trying to do in acute kidney injury. Now, one of the ways to enrich is to think about, and again, what we're trying to do potentially is enrich for this group, maybe as far down as this group. I don't think we want to enroll clinical trials to try to prevent this group of patients. So how do we identify this group of patients? One way is to use biomarkers, as has been suggested by Claudio. Here's a study looking at a variety of different biomarkers. This was done before NephroCheck, but there's a variety of different markers on this slide, all of which seem to have some discrimination, some better than others. Let me just focus on one of those. This is urine NGAL, and this is probably about the worst performance of urine NGAL ever shown. Most of the studies are a bit better than this, but let's just say it's this bad. This is 0.68 is the area under the curve. for detecting rifle criteria within 24 hours. And you might say, well, I can't use that for enrichment. It's not good enough. But is that really true? If I look at the median right here, I have a scenario where I'm differentiating patients with a 1 in 8 risk of acute kidney injury versus 1 in 35 risk. Are you really prepared to tell me that you would rather enroll these patients compared to these patients in your clinical trial? Are you prepared to tell me that enrolling these patients wouldn't save me a lot of money compared to enrolling these patients? If you know anything about clinical trials, you know that the 3 rules of clinical trial, 3 rules of real estate are location, location, location. The 3 rules of clinical trials are event rate, event rate, event rate. You want this event rate. You don't want this event rate. The problem with NGAL is actually that the event rate doesn't get much better as I go up, and I can't really find an event rate that's high enough to really be successful. But there are some biomarkers that can get us there. So what would I use biomarkers for? Well, I use them for entry criteria, as I suggested, by enriching, excluding low-risk patients, narrowing the effect, excludes cases where the drug can't help. I'll get to that in a little bit more, but it involves patients that have the wrong etiology, patients where injuries already occurred. If I have a preclinical study that shows that I can only affect AKI if I can get ahead of it, then I might use biomarkers to exclude patients that already have the disease and prevent the disease in patients that haven't manifested yet. It can also be used, and Claudio said this at the end of his talk, potentially as part of the endpoint. Remember, much of AKI, particularly in relatively healthy patients, is subclinical. And can I increase my endpoint detection by using biomarkers? Can I improve my adjudication accuracy? And finally, can I improve the risk-benefit ratio by avoiding the exposure of low-risk patients to the adverse effects of the intervention that I use? So let's put some numbers on this. So if we imagine a new drug that has a 20% relative risk reduction, and that's about right for a drug. I'm not going to really achieve a drug that has a higher relative risk reduction than about 20%. If my event rate is 50% in the placebo group, and of course that would require some degree of enrichment, as I just said, I would be treating, I would bring that down to an event rate of 40% in the treated group. That's a 20% relative risk reduction. That's a 10% absolute risk reduction, which means I would need about 776 patients in my trial. If I charge $12,000 per, and that's about par in the US for what it costs to conduct a clinical trial, is about $12,000 per patient. That's about $9 million. Okay, that's what it costs to do a trial of 776 patients. Wonder if I could do a few things that are a bit different. Wonder if I could take that and convert it to an enrichment strategy by restricting to patients that have more than 50% risk, by eliminating the patients that have a lower than 50% risk. I still have a 20% relative risk reduction because that's the drug, okay? I can't change that. But now my absolute risk reduction goes up to 13.5%. Well, does that have any effect? That's not much of a difference, right? And has a huge effect. My sample sizes drop to 430 patients. What does that do to my bottom line? My trial now is $4 million less. I'll split it with you. Give me $2 million, you can have the other $2 million. So it's a significant cost savings. Wonder if I can take out 50% of patients, wonder if I can have a scenario where because 50% of the patients may have zero risk of AKI in many situations, cardiac surgery is an example. Many patients have very, very low risk. Wonder if I could remove those patients. Well, here again, I'm maintaining if these patients are in, I have a 20% relative risk reduction, but now my absolute risk reduction is decreased to 6.3%, and my sample size balloons to 3,000, and my cost goes up to 36 million. If I can take those patients out, I can get back down to a more affordable trial. Wonder if I take 50% patients who can't be helped. The same thing is true. Now my absolute risk reduction falls to 50% and my trial size increases even more. How about if 10% of the patients already had the endpoint? Well, now I'm actually affecting both relative risk and absolute risk because now the drug can't work because the patients already had the disease. And now, so my relative risk reduction drops to 16%, my absolute risk reduction falls to 8.8%, my trial costs balloon up. Again, if I can use a biomarker to convert back to a more favorable state, I can reduce costs. How about if I try that? How about if I take 10% who've already had the endpoint and I have a biomarker that excludes 100% of those? Well, I could go down from my cost of $12 million all the way down to $9.6 million. So I can save $3 million on the cost of the trial by eliminating those patients. Wonder if my biomarker isn't that accurate. What if it only excludes 66% of patients who already have AKI? I could still get my absolute risk reduction down to 9.4% or up to 9.4%, and my costs down almost $2 million. Even if I have a biomarker that's worse than any biomarker that's ever been described in the literature as far as I know, I can still save $1 million on my clinical trial. You can't afford not to use biomarker enrichment in clinical trials. It's amazing that, and the main reason people don't do it is they're concerned, and rightfully so, is they're concerned that their label is going to then require the biomarker. But I submit to you that the cost of a negative trial is far in excess of the cost of narrowing your market size by the amounts that we're talking about. How about if 10 to 20% of the events are missed? Can I bring those events? Now I'm talking about a situation where the patients benefited from the therapy, but I miss the event, and I'm missing the event in the placebo group, and so I can no longer see the benefit. So I think my event rate's 50%, but it's really 55% because I'm missing 10%. Wonder if it's 60%. If I can use a biomarker to capture those, I can show a much larger effect size. It's still a relative risk reduction is still the same because that's intrinsic to the therapy, but I can increase the absolute risk reduction from 10% up to 11% or 12%. I can decrease my sample size down to 648 or 540 patients. I can save, again, a couple $1 million or $3 million if, in fact, I can reduce it even that much. Wonder about a situation where the biomarker increases the events by 30%, but either 0, 10, or even 30% of those are false positives, because you might be thinking, well, wait a minute, if I increase this event rate detection, surely I'm not going to be accurate all the time. Wonder if I have a 30% false positive rate, I still win. I go from a cost of the trial of $9 million down to a cost of just over $6 million. So I still save almost $3 million on the trial. Now you might think, well, I don't run a big biotech company. I don't care about the cost of clinical trials. The problem is you want to use the drugs that the clinical trials will demonstrate, and we can't do that if we keep doing trials where we just fire at everybody and hope to hit something. What about the cell cycle arrest biomarkers? Do they work any better than some of the older biomarkers? Well, they do, actually, and the main advantage is shown on this slide. This is urine NGAL, and you can see there's a very clear increase in NGAL as a result of going from no AKI, stage 1, stage 2, stage 3 AKI. It's clear that the biomarker works, but there's a lot of noise. These are conditions where there isn't AKI, and the biomarker is still sort of up in this range. This is Nephrocheck. Again, it works. Doesn't seem to work that much better than NGAL over here, but look at the noise. It's way down. It's way down. This is like Bose noise-canceling headphones. It's still the same music, okay? But now I filtered out all of the rest of the noise. In fact, we've done some recent work showing that NGAL and Nephrocheck actually correlate very tightly. There's very few situations where NGAL, when you have AKI, there's very few situations where Nephrocheck is positive and NGAL is negative, but there are lots of situations where NGAL is positive and Nephrocheck is negative and the patient doesn't have or doesn't develop AKI. What about the use of the biomarker in clinical trials? Well, Alex Zarbach has shown that there's a very nice kinetic around cardiac surgery, and it goes up within 4 hours of cardiopulmonary bypass. In fact, we've done some more recent work, and others have done, showing that this goes up even faster. Alex didn't measure before 4 hours, but it's already elevated quite nicely. The performance in cardiac surgery is quite strong. Area under the receiver operator characteristic curve is 0.86 for this population. The relative risk around the 0.3 cutoff is 7. And he's shown in a clinical trial that he can use this to, and this trial has been shown multiple times here as just an illustration of how this works. And probably so the intervention is shown here. This is a protocol for fluid optimization. But what I want to emphasize for the purposes of this talk is that he excluded a third of the patients, or 2/3 of the patients, who didn't have. He started out with about 900 patients. He excluded 600 in which didn't have risk high enough to be appropriate for the trial. So he got rid of those low-risk patients, and now by an enriching to this high level of AKI, 71% for any AKI, 45%, 44.9% for stage 2-3 AKI, he could clearly show benefit from his intervention. Had he enrolled all those low-risk patients, do you think the K-Eagle bundle would have been effective? No. So the trial would have been a bust. If this was an experimental agent, it would have been killed. If you're trying to do a quality improvement exercise in your hospital, you'd be told stop. So you want to enrich, whether it's for doing clinical trials or patient care. And that's what we tried to emphasize in this editorial that's been showed. Ivan Ghosh presented his study, which is in non-cardiac surgery patients, and he showed exactly the same thing. The interventions was a little different. It was a nephrology consult, medication review, increased attention to fluid management. And what he was clearly able to show was there was a reduction in the length of stay. Unfortunately, he chose any AKI as his primary endpoint, and he just missed it. But his secondary endpoint, stage 2-3 AKI, was clearly statistically significantly decreased, despite the fact that this was a very small study. So now we have 2 trials showing that enrichment using a biomarker and executing a protocol results in a reduction in AKI. So in conclusion, AKI is extremely common and is associated with increased costs and worse outcomes. However, the etiology is usually multifactorial. It's sepsis, CPB, hemodynamics, cardiac dysfunction, nephrotoxins. Timing is not always predictable, and severity is not always clear. TIMP2 and IGF-BP7 in the urine are robust measures of AKI manifesting in 12 to 24 hours. Patient selection, high versus low risk, will be necessary for effective prevention, early management strategies to work, and they're absolutely essential for us to make progress in clinical trials. And with that, I thank you for your attention.